Good morning, everyone. We are the St. Stephen's College iGEM team, and today I'll introduce what our project is about, which is the biosynthesis of velar using cyanobacteria strain Synechococcus elongatus UTEX 2973. So first I'll introduce the background of our project. So what are lactams? Actually, lactams are a family of cyclic amides, which are important commodity chemicals used in industrial production, which include nylons, antibiotics, and components in drugs. But for our project, we mainly focuses on the production of velar, which is a kind of 5-carbon lactam, as you can see. And the produced velar can be processed into piperidines used in drugs or other types of nylons, for example, nylon 5. So why do we actually need a biosynthetic way to produce these velar? Actually, various sources of lactams are produced through petrochemical sources traditionally, but what is not ideal about the petrochemical synthesis is that it requires high temperature and also harsh acidic conditions, and that resulted to a large amount of waste salt. And these are not ideal for our environment and brings a lot of harm to our environment. But on the other side, the biosynthesis alternative can provide a more sustainable and greener way for us to produce these lactams, since the chemical reactions can be carried out in milder conditions with lower pressure and lower temperature, and it can also bypass the use of harsh chemicals and non-renewable chemicals. And to mention that our team will use the cyanobacteria strain Synechococcus elongatus UTEX 2973 as a chassis for the biological synthesis of velar, since it comes with a few advantages, and our member will mention it later. And to emphasize again, our aim is to provide a biosynthesis way of velar using cyanobacteria strain Synechococcus, and this can help to develop a sustainable and more green alternative to replace the traditional method, so we can reduce the harm to our environment while also coping with the demand of velar. And what will be included in this presentation is that Halim will introduce what is involved in the synthesis pathway and how will cyanobacteria be engineered to produce velar, and we'll also talk about what impacts does our project have on the community and how can we improve. And now I'll pass to Halim to talk about the design.
So I'm Helene, and now I will talk about the design. So here we have the overall biosynthesis pathway for velar. It first starts off with carbon as the sole carbon source, and then through the biological pathway in the cyanobacterial strain, there will be production of lysine, which will then be converted to 5-ABA and then to velar by a few enzymes. So as it's first said, lysine is needed, and work from Courage demonstrated how they successfully achieved overproduction of lysine with carbon dioxide as a sole carbon source in the cyanobacterial strain Synechococcus PCC 7002. So through some research, we found this other cyanobacterial strain Synechococcus UTEX 2973, which is closely related to the PCC Courage used. They're actually same species but just different strain. But for UTEX, it is more strongly growing. It has higher tolerance to temperature and illumination, which makes it more flexible when handling it. And it has the quickest measured growing rate among cyanobacteria for like a doubling time of about 1.9 hours. So this shows that it is a promising for the synthetic cell factory we can use. Besides that, various biological tools and successful engineering of this strain to produce other biomaterials has also been demonstrated, which shows that we have a strong foundation to work on for our project. And for the other biosynthesis work for synthesis of lactams, researchers have used heterotrophic microorganisms, for example, like yeast or bacteria, but has not been reported work on photosynthetic microorganisms. So this is why we wish to use a cyanobacteria strain to provide a sustainable approach to the synthesis of valeriolactam. So back to the lysine overproduction I mentioned before. Courage, they demonstrated how aspartate kinase, or AK for short, this enzyme, is a key in the biosynthesis of lysine in the cyanobacteria. So as you can see in this flow, here is shown a part of the lysine synthesis pathway. So the AK, it actually converts L-aspartate to L-aspartyl phosphate, which will then be further converted to lysine in other processes. So it's shown that there's this AK enzyme, it is subject to feedback inhibition by lysine, which means if there are high levels of lysine production, then it will suppress the activity of the AK. So then you will have lower production of lysine. And if we want to use lysine as a substrate to produce valeriolactam, we need to bypass this regulation. So we want overproduction. And basically, they demonstrated how by introducing a feedback-insensitive AK variant, so we can remove the inhibition of lysine to the AK. So that means the AK will continue to facilitate this lysine production, but high levels of lysine will not suppress its activity. So basically, when there are high levels of lysine, you will continue to have the production, and so therefore we'll have overproduction of lysine. And that is done by a specific point mutation in the AK, and we will insert this AK variant, the sequence of this AK variant, into the genome of Synechococcus, so there will be overproduction of lysine. So after we have achieved elevated production of lysine, we'll then need to convert it to 5-ABA. And 5-ABA is 5-aminovaleric acid, and it is the lactam precursor of valeriolactam. This acid is naturally produced from the 5-aminovalerate pathway, which involves two enzymes from the bacteria Pseudomonas putita. These two enzymes are the L-lysine monooxygenase, or DAVE-B, which converts lysine to 5-aminovaleramide, an intermediate in this reaction, and DAVE-A, which catalyzes the 5-aminovaleramide to 5-aminovaleric acid. And so researchers have used this pathway in yeast and bacteria to produce 5-ABA, and so now we are trying it out in Synechococcus. And similarly to AK, the gene sequences of these two enzymes will be inserted into the genome of Synechococcus. And finally, we need to cyclize the 5-ABA to valeriolactam. So this is done by a cyclization enzyme, and previous reports on the cyclases show that they lack efficiency. So we are remodeling, optimizing a known cyclase to try to increase its selectivity and activity through modeling. But today I will not go through in detail, so you just need to remember a cyclase will cyclize the 5-ABA to valeriolactam. So basically, in summary, to recap, carbon dioxide will be used as the carbon source, and there will be production of lysine, and it will be converted to 5D and then valorolactone. So now you may be wondering how we will establish this valorolactone synthesis pathway in Synechococcus. So as I mentioned before, we need to insert the genes into its genome, and through that we will achieve that by using a system called integrate system, and we also subclone that system into a shuttle vector designed by iGEM team Marburg 2019, which I will go through later. So for the integrate system, it is actually a CRISPR transposon system that, as you can see here, is an illustration of how the protein complex looks like. There's a piece of red CRISPR RNA which directs the protein complex to a designated site in the genome of an organism, and then the protein complex will insert a piece of DNA into the genome. So the plasmid for this that has the genes for this CRISPR transposon system is called pSpin, and basically we need to engineer this plasmid so that it will work in our host organism. So first of all, besides inserting the genes of interest in this plasmid, we also need a promoter that works for expression in cyanobacteria, and also a site in the genome of Synechococcus for the DNA insertion. So a set of biological tools for the use in Synechococcus has been provided by Lee and others, and these include strong promoters. So we selected PBSBA3 and PCBC560, these two promoters for the expression of the CRISPR transposon system and also our genes of interest, because they show high transcription levels. And for the site of DNA insertion, it has been identified that there are neutral sites in the chromosome of Synechococcus which are suitable for DNA insertion. So our CRISPR RNA will be targeting a neutral site. So our genes of interest will be inserted into a downstream of this designated target site in the genome by the CRISPR transposon system. And we will have to subclone the entire CRISPR transposon system into a shuttle vector. So this shuttle vector is based from iGEM team Marburg 2019, and it has the origin of replications of Synechococcus and E. coli, which means it can be replicated in both of these organisms. So actually, why do we need this shuttle vector? It is because for the CRISPR transposon system, the pSpin plasmid, it does not have the origin of replication for Synechococcus, which means if we just directly transform that plasmid into Synechococcus, it will not be replicated in our host organism. So that's why we use this shuttle vector, so it can be replicated in E. coli for plasmid preparation, amplifying enough amounts of plasmids. And then we will use those plasmids and electroporate it into Synechococcus for gene expression. And we also modified this shuttle vector to serve our own personal needs, which include we added a multiple clone site in their shuttle vectors, and this is where we will subclone the CRISPR transposon system I mentioned before. So to recap of what I just said, basically we will use the CRISPR transposon system and target our genes of interest into the genome of Synechococcus. And to do that, we also need to use the shuttle vector so that the plasmid can be amplified in E. coli and then expressed in Synechococcus.
So it's important that our project does not only work in the lab but also in real life. So for the human practice part, we visited and interviewed two companies, and one of them is the JAB Impact Technology. So JAB Impact Technologies mainly focuses on the algae cultivation for production of raw materials, which can be used as a food ingredients. They work in algae is similar to ours in cyanobacteria, since both of these organisms have photosynthetic abilities. So the techniques to cultivating them share a lot in common. So besides cultivating techniques, the goal of the work is to provide a green alternative for industry. So we visited them to learn about the impact of these technologies and what this technology has to the environment and the community, and also the challenges we may overcome when we are cultivating the algae. So the representative of JAB Impact mentioned that how using algae for production of material has the advantages of lack of the use and release of toxic substances, as it uses photosynthesis, in which carbon dioxide is used for synthesis of biomaterials. It can also help fight climate change too. So these characters help make algae technologies a sustainable alternative to traditional production of food ingredients, which can help improve the food security in the community. So JAB Impact also mentioned that how costs and techniques to maintain growth conditions such as the suitable growth temperature and contamination when cultivating the algae. And these are some major problems and challenges in these technologies. So from our visit to the JAB Impact, we could see that the selecting of cyanobacterial strain Staphylococcus was proper. But it is promising in providing sustainable alternative. Companies like JAB Impact which hope to achieve, and its high tolerance also allows flexibility when handling them. However, we also learned that we can improve our project by improving techniques to upscale the growth of Staphylococcus and also maintain its purity. So though the company are mainly focusing on the cyanobacteria, but the techniques are still worth learning. So besides of JAB Impact Technology, we also interviewed another company called Hong Kong Research Institute of Textile and Apparel Limited. So they focuses on supporting development and marketing of innovation in textile technologies. For example, designing new textile materials and developing methods to recycle garments. As the villarolactam produced in our projects can be processed into nylon, we recognize that the textile industry may be part of our products and potential end users. So we arranged an interview with them to know about the market and consumer in this textile industry. So they pointed out that there is a big demand of sustainable production of nylons. They also mentioned how industry have started to use life cycle assessment to measure how effective the product is to the environment. So this shows that the textile products and industry are practicing methods to become greener. So as for the customers, more customers have more aware about the sustainability, not only in Hong Kong but all around the world. However, there is still a lack of motivation from customers to push green products, and the main driving force of green technologies still come with regulations and governance requiring industry to become more green. So from the interview, we learned that our project is in line with the demands of the market as it provides a green alternative for the step of producing lactam of nylon. So we also can promote green technologies among the public so they are aware of the sustainability issue and know how research and industry of green alternative can improve the environmental issues.
So to summarize it all, we aim to use Staphylococcus to provide a biosynthesis pathway for villarolactam, which will replace petrochemical synthesis of lactams. And by this, we will use carbon dioxide as the carbon source to convert it into villarolactam. And to achieve that, we use a CRISPR transposon system to engineer the genome of Staphylococcus, and we'll be also using a shuttle vector to aid the cloning and expression processes. So that's the end of our presentation. So questions? Yes.
It sounds a bit confusing for me that you are trying to use a more eco-friendly way to produce synthetic fiber. especially for mass production, is I imagine is very energy consuming than E-coli, for example. So yeah.
Okay, so for your first question about why we choose to produce lactams. So actually, nylons, it is, like there is, as I mentioned, like there's demand for a sustainable approach to produce nylons, which this actually means like now investors, they are interested, because nylon is a type of textile fiber that is really, there's a large demand because it has a lot of flexible uses, and so investors are looking for a way, like a greener way to produce these. But for our project, it provides like it for lactam is one of the steps for producing nylons. So it's actually like we are providing a green way for this step for nylons. But actually, besides nylons, as I before we also mentioned that lactams is not only used for nylons, it could also be used for raw materials for components in drugs and other like materials. So we are just like trying to make this the production of this raw material a greener way. And for the comparison of other bacteria and with cyanobacteria. So for bacteria, you need to like feed it other you need to feed like sucrose or for it to grow, right? But for cyanobacteria, we it is true that the growth conditions need to be maintained, like for optimal growth. But so we wish to like from our interview with Jeff Impact, we know that we in our project we also need to develop techniques for better growth of this cyanobacteria. So that way, as climate change has become a more popular topic in the society, we can try to use this cyanobacteria to also help take up the carbon dioxide in the atmosphere. So I hope that answers your questions. Yes.
May I ask how much cheaper for your synthetic routes for making the lactam?
What?
How much cheaper for your synthetic routes for the lactam? Like, you said original synthetic routes they use toxic chemical, and now you use CO2, but at the end, how much do you cost to make a lactam?
So the cost is mainly something like in research. First of all, in for like the whole process of you can say like a product. So now our work is still in the proof of concept stage, like in the lab. So we are still we don't have that much results yet. So it is still difficult for us to evaluate, like to measure how much energy we have used for this work, because we are still in the stage of lab research. But if this if this can be scaled up to pre-initial scale, then for that we can use, like I said, life cycle assessment to evaluate how effective our work actually is, and how much cost we're using. So to be honest, right now it is hard to say about the cost. Yes.
I would like to ask about the AK, the kinase that you talked about. So you mentioned that it has a negative feedback loop. Is it an allosteric feedback, or is it a gene expression kind of? Like where does the feedback affect? Does it affect the expression of the AK, or does it affect the activity of the AK?
It affects the activity of the AK. So it kind of like suppresses like the activity, so there won't be as much lysine that can be produced. But if you're interested in this in the cycle, you can take a look at this paper which we referenced it on, and it explains in more detail of the cycle.
Because I was just wondering whether you've locked up the endogenous
we are just introducing feedback insensitive AK. So while the wild type is being suppressed, we have this AK variant that will keep the lysine production going. Any more questions?
Yes, here's another follow-up question from Anna. So, in my point of view, I don't see any problems in producing nylon because as long as the source is clean, just like what you're saying, I think it's fine. But I think Hannah's point is the algae you choose to produce the raw material actually needs huge amount of energy to maintain for their growth and survival, and also the production of the enzymes to produce the lectin. And because I think you aim for mass production, right? Your project, manufacturing.
Yes, it is a consideration in like the later stages.
Yeah, so in my point of view, I think you might need to have a better rationale for the chassis you choose. And what's my second question? Oh, yeah. And because you're aiming for manufacturing, do you have any ideas how to purify and isolate the lectin you produce?
Yes, so for the downstream analysis, we will use like a liquid, like a solvent test to first because valeriolactam it is soluble in carbon tetrachloride, and so after we obtain the result, and then we will dissolve everything in this solvent, and then through liquid extraction, and then we will have to quantify the valeriolactam. And besides the valeriolactam, we also can quantify the amount of lysine and 5-ABA. So through another test, Hinsberg test. And this, because now we are still in the lab stage, so quantifying these will also help us know and which step in the production we need to increase so there's better production, better quality.
Very